Hi, hello. Welcome to the Physionic podcast, or welcome back to the Physionic podcast if you're a long-time listener or viewer. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and I suppose particular to this particular study that we're going to be looking at, I am involved. I actually do research in mitochondria, mitochondrial dynamics, and autophagy and apoptosis, but really the first three are going to be the ones that we're going to be focused on for this particular podcast episode, wherein we will be covering a paper that I found pretty intriguing. Actually, my boss, my mentor, sent this paper to me because he thought that it might be helpful for my thesis, for my dissertation. So I went ahead and took extensive notes, and then I thought, this might actually be really interesting for other people as well. So I'm going to go ahead and be explaining some of the data on a particular paper called Lipids Activate Skeletal Muscle Mitochondrial Fission and Quality Control Networks to Induce Insulin Resistance in Humans. And if you're not familiar with what lipids are, they are fat. So fat that we consume. And this was actually published pretty recently, so less than a year ago, and it was published in the Journal of Metabolism, which is actually where I had one of my publications, was in the Journal of Metabolism. So we're going to go over, as usual, I'm going to give you a little bit of an introduction on everything. We're going to be going over how fats in the bloodstream, kind of in layman's terms, how the elevation in fats in the bloodstream, let's say after a meal, for example, how that affects our skeletal muscle, but more specifically, how it affects the mitochondria within. The amount of mitochondria, how it affects the behavior of mitochondria, and if it's degraded, do we have greater levels of autophagy or lower levels of autophagy as a result? So things like that, all those things are going to be covered in this particular podcast. And then I may do a follow-up podcast on this paper, also looking at insulin resistance, which is the other half of this particular study. But for this podcast, we're going to be focused on its effect on mitochondria. And there's some pretty interesting results here. So I'll be explaining the mechanisms and whatnot as well as we go through. So a bit of introduction. So the researchers, this is an introduction based off of what the researchers pointed out, is that people with diabetes tend to have mitochondrial abnormalities. And those abnormalities can come in a variety of different ways, but one of them is by the shape of the mitochondria themselves. So metabolism, so if your cells are using fat, or if they're using glucose, for example, blood sugar, for energy, change the look and shape of mitochondria. And this change in shape may be related to insulin resistance. So that's kind of like what we're alluding to potentially another podcast. And let me know if you're interested in having another podcast. I guess before I go any further, I usually have my disclaimer of, if you're just listening to the podcast, have no fear. I'll be explaining things, but it might be good to hop on over to the video version at some point, because I'm going to be showing a little bit of data, but I'm going to be showing images, some actual microscope images of mitochondria and autophagy vesicles and things like that. So if you're interested in seeing that, then hop on over. But back to the introduction. So mitochondria are constantly undergoing changes in their shape, and the shape is kind of considered mitochondrial dynamics. So the changing of shape to becoming really elongated, or they can become really fractured and can become really small and kind of circular. And that happens through a process known as mitochondrial fission and fusion. And when we're talking about fission, we're talking about the separation of mitochondria. I don't have the graphic that I thought that I had, unfortunately, but you have a single mitochondrion, and then it gets split down the middle. It actually gets squeezed together until it pinches, and then you have two new mitochondria, or one new mitochondria from the original. And that is mediated by a particular protein, and I'll go over this again, by a particular protein called DRP. it's dynamin-related protein. You don't need to know the name, just know DRP. But again, I'll reiterate things. Then fusion, on the other hand, is the combination of two mitochondria into a single mitochondrion, so a larger mitochondrion. And this is mediated by a process called fusion, and this is by a particular protein called MFN. And there are a variety of different proteins that act on both of these functions, but we're going to be focused on two of them. DRP for fission, splitting mitochondria apart, and fusion, which is the combination of mitochondria into one, and that is mediated by MFN, or the full name is mitofusin. Mitofusin 1 and 2 are the two isoforms, or different versions of that protein. But MFN, that's what you need to know. So it seems that, based off of other studies, that fats seem to increase mitochondrial fission in models of insulin resistance. So you get this association of greater fission, more splitting apart of these mitochondria lead to, or are associated, I should very much correct myself there, do not necessarily lead to, but are associated with higher incidence of insulin resistance or lower insulin sensitivity. So the aim of this particular study was to see how fat in the bloodstream affects mitochondrial behavior, as I mentioned, mitochondrial dynamics. So a bit on the study design, and then we'll go into the data, and I'm going to show you a few images. Again, if you're listening, I'll try to explain things as best as I can so you can kind of create a visual in your head. But for those on the video, I'll have a few images that I'll throw up. And so the study design was 19 participants, slightly more men. I think they had 11 men and 8 women, and that were mildly active. So I think they were active maybe 2 times a week or less, something along those lines. So not supremely active or anything like that. But they were healthy, so they had blood markers checked and everything. They had no history of diabetes. And these individuals were then recruited for the study. The study was a crossover design, meaning that every single participant that ends up undergoing one condition of the experiment ends up also going through the other condition of the experiment. So they allow a washout period of, let's say, several weeks before they retest the same individuals but in a different condition. And there were 2 conditions. So the first condition was a fat infusion condition, meaning that the researchers literally infused fat into the bloodstream for a 12-hour period, and then they wanted to see what changes would occur once they plucked out cells from these people's musculature. They took cells kind of in the midway point and wanted to then see if there are going to be any changes. Any changes ended up occurring. So that was the fat infusion condition. So if you see that, or if I mention that, fat infusion, lipid infusion, those are the exact same thing. Lipid, fat, synonymous. And then the other condition was a control condition where they just infused saline. So they essentially use the substance that the fat may have been in, but they don't actually add the fat. So you've got our fat infusion for 12 hours, and you have our saline infusion for 12 hours. And again, those are separated by many weeks so that you don't get any sort of crossover effect that might occur. But ultimately, all 19 participants underwent both conditions. And so the saline condition, if I haven't mentioned it, is the control condition. That's kind of the normal condition that you wouldn't expect. You're comparing the fat condition against the saline condition. Okay, so with that out of the way, let's go ahead and touch on a little bit of the mechanisms, and then I'm going to start going into the data, and we're going to start breaking this down as we go forward. Okay, so as I mentioned, there is this protein called DRP, and DRP, if it is activated, then it will move from what's called the cytosol of the cell, which is this area around the mitochondria, but not the mitochondria themselves. So DRP is just floating around in this area inside the cell, the predominant area of the cell called the cytosol. And if it is activated a certain way, which is called a phosphorylation of a serine site, again, you don't need to know that. All you need to know is that it's activated. But I always throw in some details because sometimes some molecular biologists like to know this kind of stuff. So there is a tag that's put on this DRP protein, and then the protein will then be attracted to mitochondria. And when that occurs, it starts to create a collar around the mitochondria and starts to squeeze in and separate out one mitochondria into two. So that's fission, right? So now, keeping that in mind, they ended up looking at that. So from the muscle samples, they've taken muscle samples and now they're testing for how much of basal DRP there is and activated DRP. Now, I'm going to pose a question to you, just thinking based off of just what I just said, which one is going to be the more important one in this situation for a measure of mitochondrial fission? If you thought activated, you're correct. So if you have elevated levels of activated DRP, then presumably that would be going to the mitochondria, would be associated with mitochondria. So what they found is that basal DRP, and if you're looking at the data, all you're doing is just comparing the darkness and the thickness of the smudges. That's it, between the two conditions. Saline on the left and lipid or fat infusion on the right. And basal DRP was the exact same with both conditions. So basal DRP did not change with the infusion of fat into the bloodstream. However, activated DRP did increase. So this phosphorylated, this activated, this tagged version of DRP did increase with the fat infusion. So that gives us a small piece of information right there. Now, the other side, right, the flip side of that I mentioned in mitochondrial dynamics, how mitochondria behave in a manner of speaking, is fusion. And that is regulated by a protein, and I actually do have this graphic for you, known as MFN. And MFN allows when two mitochondria get close with one another, MFN will sort of interact with the other mitochondrion, and eventually a series of other proteins that I'm not mentioning, like OPA, for example, or MFF, or I'm sorry, MFF is actually a fission protein, but like OPA, for example, those proteins end up also getting engaged, but we're focused on this mitofusin protein, this MFN protein. And ultimately, on the membrane of the mitochondria, this MFN protein ends up interacting with the membrane of the other mitochondria, and that ends up allowing for those two membranes to start fusing together to allow for the fusion of two mitochondria into one larger mitochondria. So, looking at the data for that, and again, we're doing the same thing. We're just comparing the smudges. The experiment is called a Western blot, incredibly popular, probably one of the most popular experiments in all of molecular biology. And what they found, the researchers found, and of course make your own assessment as you will based on your eye, but what they found is that there were no differences between the two. And they actually did quantification as well, so it's not like they're just using their eyeballs. They're actually using computer programs as well to figure out if there are any differences. And they found that there were no differences in MFN proteins. And this was also true for the other proteins as well, mitofusin 2 as well as OPA, which is the protein that I briefly mentioned earlier. So here we're seeing evidence, right, at this point that fission might increase, at least DRP levels, activate DRP is increased. Fusion, however, is unaffected by this fat infusion, this fat increase in lipids or fat molecules in the bloodstream. Okay, so then presumably you'd be thinking, okay, well, if you have more of this phosphorylated DRP or this activated DRP, then you might have greater fragmentation, more fission occurring where single mitochondria are becoming, one larger mitochondria is becoming fragmented into two different mitochondria. And that's actually exactly what they ended up finding when they actually quantified the number of fragmented mitochondria in the lipid condition, in the fat infusion condition. They saw a drastic increase in the number of fragmented mitochondria. I don't want you to necessarily think that fragmented mitochondria are bad. It's just a biological process by which the cells are just separating out single mitochondria into multiple mitochondria. And it is actually important for one, biogenesis. The creation of more mitochondria is also follows certain guidelines like this as well. So it's really very much dependent on the context on if this is a negative thing or a positive thing. So I don't want you to think the word fragment may have a negative connotation to it. So just keep that in mind. Try to make sure that you think about it within context. It's not necessarily a bad thing. Okay, so they did see greater fragmentation, and they do see some minor evidence that potentially fission is upregulated as well. So then another thing, and here we're going to start going into autophagy, and then I'm going to circle back onto mitochondria. So autophagy is, I've made a graphic for this as well. So how this works Actually, my lab just released a paper showing that there is another mechanism by which this style of autophagy can occur, which is mitophagy, the autophagy of mitochondria, the destruction of mitochondria, because autophagy is the destruction of organelles. And in this situation, it's, or not just organelles, but certainly it is a big part of the destruction of organelles. So in this case, we're talking about mitochondria. So what I've reported on this in other videos when I covered other studies as well, is that with fission, sometimes you can also get the encouragement of mitophagy, the destruction of these mitochondria. And how does that happen? Well, putting my lab's research aside for a minute, and we just focus on kind of the conventional way by which autophagy occurs, you have your mitochondria and you have your outer membrane of your mitochondrion, and there are two key enzymes that play a critical role in the regulation of mitochondria, in the allowance or disallowance of this destruction of this particular mitochondrion. And how that occurs is through these two proteins. One protein is called pink, and that's why if you're watching the podcast, I colored it as pink, and the other one is parkin. So this pink parkin system allows for the destruction of mitochondria. It also allows for the destruction of particular proteins on the mitochondrion, but we're more focused on the overall destruction of the entire mitochondrion through autophagy, called, again, mitophagy. So what pink does is it starts phosphorylating or tagging these different proteins all across the mitochondrion. And this actually also allows for another type of tag called a ubiquitination. So a ubiquitin tag. So another molecule, you have a phosphorylation tag and you have a ubiquitin tag. So the ubiquitin tag gets added to the mitochondria, and then pink will phosphorylate that ubiquitin tag. So it will add a tag to the tag, essentially. And the more that occurs, the more it recruits parkin. And parkin is a free-floating protein or enzyme that then gets recruited to that site and starts adding a lot more ubiquitin tags to the mitochondria, to different target proteins all across the entire mitochondria. But if enough of these target proteins are tagged by parkin and by pink through ubiquitination as well as through phosphorylation, then eventually you have an autophagy vesicle that will come in and start to degrade it. So let me show you a picture of that. So this vesicle called a phagosome will come in and essentially will recognize all of these ubiquitin tags all over the mitochondrion. And that's not saying that the mitochondrion needs to just get rid of one protein or needs to get rid of a few proteins. It needs to get rid of so many proteins that the cell just is like just going to get rid, evolutionarily speaking, just wants to get rid of that entire mitochondrion. So what it does is it creates this vesicle, this fatty lipid that circles all around it and literally creates a sac or a circle around the mitochondria. And then eventually that ends up going through autophagy, meaning the degradation of the entire mitochondrion. So that is roughly how it works. There's a lot more complexity. You're talking about other proteins, P97, you have LC3, you have lysosome attachment. You have a series of other processes that occur, but I'm not trying to bog this down too much. So now going back to the data, what happens when you infuse fat into the bloodstream? You see this increase in fat in the bloodstream. And what happens to Parkin and Pink first? And then we'll go and look at some of these images of some really cool images that I'll explain in just a little bit. So again, here we're just doing a comparison, and the researchers found that the levels of Parkin did not increase. So there was no change in Parkin, which is the enzyme that adds these ubiquitin tags to the mitochondria. However, there was an increase in Pink amount. So the amount of that particular enzyme was increased due to fat or lipid infusion. So this provides some evidence that maybe there is something going on here that may be leading to degradation or loss of mitochondria as a result. However, admittedly, this is weak evidence. This is very weak evidence. So, and the researchers, of course, knew that, so they decided to do a little bit more digging. They did a little more investigation. So they ended up taking images, electron microscopy images, that's what those are called, and they showed that, and hopefully I've labeled this well enough for anybody who's watching, the top two images are of the control condition, the saline condition, and the bottom two images are of the lipid infusion condition. So what you're looking at is literal images of cells or within the cells, and the white blobs, you're seeing the dark blobs. The dark blobs are mitochondria, like literal actual mitochondria. And the white blobs are these autophagy vesicles, these phagosomes. And what they noted is that there were higher levels, although the mitochondria were roughly the same between in terms of what seemed like the health of the mitochondria were roughly the same. However, there were more autophagy vesicles in the lipid infusion condition. And this is pretty subjective. I mean, you're just taking images and you're just kind of looking. And of course, there's programs and softwares to actually quantify some of this stuff. However, I will admit that they did not do any of that. They didn't do any sort of quantification on that. So we're just looking at the actual images. And like I said, the lipid infusion did show higher levels of these autophagy vesicles, while by comparison to the saline condition, which didn't see any really, or really small amounts. So this all leads us to potentially think maybe mitochondria are being degraded. Maybe there's more autophagy going on, more mitophagy going on when the blood is filled with more fats in it. However, contrary to that thought process, they did measure the total amount of mitochondria in the cells, and they found there was no difference between the two conditions. So, no statistical difference between the two conditions. So, mitochondria, the total amount of mitochondrial content in the saline condition was the exact same as the lipid condition. So, that is extremely interesting because that implies that mitochondria are undergoing fission, most likely, but because they're fragmenting, but they are not being degraded in this situation. So the overall content is the same. So the behavior of mitochondria, quote unquote, behavior of mitochondria is changing, but the content of the mitochondria is not changing. Okay, so there's the data. That's what I've got for you. However, let me put a nice bow on this. Let's do conclusions and kind of the takeaways, really more so the conclusions in this situation. The data, and I do have one more point that I wanted to add to this, which I think is important. This data shows that there's an increase in fragmentation of mitochondria with exposure of high levels of fat in the bloodstream. Okay, that fat is a combination of different fats. However, it is predominantly polyunsaturated fats. So I'm sure the people that are all anti-polyunsaturated fats are just looking at the chops right now, just loving this right now, hearing that. But that is what they used. They used a system that was predetermined, like the amount of all these different fats. And from what I remember, I believe it was polyunsaturated fats was the major fat that they ended up using. There were saturated fats, I think, as well and whatnot, but the big one was, like I said, polyunsaturated. So this is likely due, this fragmentation of mitochondria is likely due to increases in fission. However, mitochondrial content itself is unaffected. Now, here's my point. Why might that be? Well, it might be that the exposure to fats was not long enough. That 12 hours, or when they did their collection, wasn't long enough. You were just starting to get some of this initiation of this degradation, machinery starting to build up. You were just starting to get some of these, you getting an increase in pink protein, maybe down the line, maybe 4 hours later or 6 hours later or 2 days later, they would have seen increases in parkin. They would have seen more autophagy vesicles. They would have started seeing decreases in mitochondrial content, but that is mere speculation. So we don't actually have any evidence of that. I'm just guessing that if they had done the experiment for maybe a day or maybe a week of just continuous fat infusions, that maybe they would have started to actually see an effect on the mitochondrial content. So the mitochondrial content did not change, however, based off of a 12-hour fat infusion. Now, I did think that this evidence is not overly convincing on its own. I think we certainly need more studies. I don't think it's that straightforward. I did not like that they could have done a lot of microscopy experiments that would have been far better off. So I'm talking like, so in our lab, for example, we do a lot of fluorescent microscopy. It would have been great to see this phosphorylated DRP, the protein that's responsible for fission, if that had actually moved to the mitochondria and see it associated to the mitochondria. That would have been great to see. They could have done some further experiments to really figure out if there were real changes in fission and fusion, but this is kind of like a step in this direction of just kind of figuring it out. And of course, like I mentioned, having it longer. However, unlike other people that are just like, oh, this study's worthless because they only did it for 12 hours or they did it for 12 hours. That's ridiculous. Everybody always has a complaint. That's true of every single study. They have to have a stopping point. They can't just continue piling stuff into a single study. They have to just publish what they have, and then they go from there and maybe, try different conditions. Like maybe next time they'll do 24 hours or 5 days or something along those lines. Maybe they will do fluorescent microscopy. Maybe they didn't have the capability at that point. Like there are a number of different ways. So I'm not at all like saying that they did a bad job by any means. I just think that they definitely need more there to actually convince me of what they're actually showing. But preliminarily, it does seem to be pointed towards greater fission of mitochondria with this exposure to high levels of fat in the bloodstream, and that may lead to mitochondrial content decline. But overall, just based on this data, there was no decline in mitochondrial content. Okay, folks, that's what I've got for you. Hopefully you found this informative, and let me know if you'd like another one on the insulin sensitivity aspect of this study, and maybe a little bit more on the mitochondria as well. And with that, I hope you have a wonderful day, and I'll catch you in the next one. See ya.